Welcome to the deep dive. You've handed us this massive collection of high-yield medical resources. We're talking notes, exam questions, core concepts across biology, chemistry, and physics. And our mission today is to distill all of that into the absolute essentials.
That's exactly right. And if you're trying to accelerate your learning, whether you're a medical student or, a self-taught learner, you know the human body isn't neatly divided by subject.
Right, it's all one integrated system.
It is. You have to understand how the physics of pressure dictates blood flow, how chemistry is driving nerve impulses, and how anatomy provides the structure for all of it. We're here to deliver that high-yield scientific literacy you need to build your foundation.
Okay, let's unpack this integration. Where do we start? Let's go with the absolute foundational building blocks, structure, and, of course, the specialized terminology.
Anatomical terminology, yeah. It can be daunting at first, but it really does follow rules. As the sources remind us, the difficulty isn't really in comprehension, it's the sheer quantity.
It's a volume game.
It is. I mean, if you see a muscle called extensor carpi radialis longus, it sounds incredibly complex. But it's just descriptive. It means it's the muscle that extends.
The wrist, the carpi.
Exactly. It lies near the radial bone, and it's the longest of two muscles. So learning the language is really the first step to fluency.
And from that language, we go to the cell level and then immediately into the organization of tissues. We've got the four primary types: muscle, nervous, connective, and epithelial. What's the big functional distinction we need to grab onto right away?
I'd say it's how the cells are packed. So take epithelial tissue. This forms your barriers, like the epidermis or the lining of glands. Its cells are characterized by being extremely closely packed together.
Like bricks in a wall.
A perfect analogy. Think of keratinocytes in your skin. They form this tight protective seal.
Okay, so if epithelial tissue is that tightly packed barrier, then connective tissue has to be the opposite, right? The structural matrix providing support and, well, space.
Exactly. Connective tissue, or CT, is the inverse. Its cells, like chondrocytes in cartilage, are widely spaced. They're separated by a large amount of extracellular matrix, what we often call ground substance.
And that space is the whole point.
It's crucial. CT needs flexibility or movement or storage. Whether you're talking about the fluid matrix of blood or the rigid matrix of bone, that space is key.
This structural idea takes us right inside the cell, to the plasma membrane, the ultimate gatekeeper. We know it's mainly a phospholipid bilayer, but what are the other components that actually let it do its job?
Well, you have cholesterol for stability and, critically, a huge variety of integral and peripheral proteins. Those proteins are the real machinery.
And a quick exam note here from the source material. The endoplasmic reticulum is an organelle inside the cell. It is absolutely not part of the plasma membrane itself.
A very common mistake to make. And those integral proteins you mentioned, they facilitate transport. We usually categorize that transport based on whether it uses energy. So first, you have passive transport. That's diffusion for solutes and osmosis for water. This movement just follows the concentration gradient. It's all downhill, requiring 0 ATP energy.
It's just nature balancing things out.
Pretty much. But the moment you need to move something uphill, against that gradient, you need active transport. And this is where the physics of flow meets the chemistry of energy.
And we're talking about the famous Na plus K plus ATPase pump.
That's the one. It requires ATP to actively move 3 sodium ions out of the cell for every 2 potassium ions it brings in. This is so critical because the pump is constantly working to create this really steep concentration gradient for sodium.
And it's the collapse of that gradient that powers everything.
Exactly. The sudden passive rush of sodium back into the cell through those protein channels is what creates the action potential. It's the basis for all nerve and muscle function.
The sheer energy that pump uses is just staggering. And since the pump itself is a protein, let's zoom out a bit and look at the chemistry that dictates how these essential structures function. Let's talk macromolecules, starting with proteins.
Proteins are built from amino acids. And when we talk about their structure, we have primary, secondary, and tertiary levels. The primary structure, that's just the sequence of amino acids, is held together by incredibly strong peptide bonds, covalent bonds.
Wait, so if those bonds are so strong, what's actually happening during denaturation? When a protein unravels from heat or extreme pH, it sounds like the whole thing is destroyed.
But that's where the distinction is vital. Denaturation disrupts the weaker, more fragile structures. The secondary, like a helix, and the tertiary, the overall folding.
And those are held by weaker hydrogen bonds.
Mostly, yes. So because the primary sequence is locked in by those strong covalent peptide bonds, denaturation does not affect the primary structure. The sequence is still there, even if the shape, and therefore the function, is totally lost.
That's a great insight. Okay, moving quickly through the others. Carbohydrates. They're only absorbable once they're broken down into their basic units, their monosaccharides, like glucose.
And for lipids, just remember triglycerols are your common dietary fats, but steroids like cholesterol, estrogen, testosterone are all derived from lipids. But crucially, coenzymes are not lipids. That's a common point of confusion. They're usually derived from vitamins.
All right, now let's pivot to the body's environment. This huge aqueous chemical ocean. Fluid balance is all chemistry, and we divide the water into intracellular fluid, ICF, and extracellular fluid, ECF.
The ECF is everything outside the cells, so your blood plasma, the interstitial fluid between cells, all of that. But a key fact to remember is that the vast majority of your body's total water is actually inside the cells in the ICF compartment.
And the gradients that NO plus K plus pump works so hard to create define the ion distribution between these two spaces.
Absolutely. ICF is high in potassium, K plus ECF. ECF is high in sodium, Na plus Ot. So this means if you find too little sodium in the blood, which is ECF, we call that hyponatremia, too little potassium in the blood is hypokalemia.
And maintaining that precise balance is like homeostatic ground zero.
It really is. It relies on hormones, the kidneys. It's a constant active process.
This leads us naturally into osmolarity and tonicity, which ties the chemistry to the physics of water movement. We need to be super clear on the difference between a mole and an osmole.
Okay, so the mole is just a unit for the amount of a substance, a specific number of molecules. But the osmole is the unit for the number of dissolved solute particles that can actually exert osmotic pressure.
And that distinction is everything from medical calculations. Give us the classic example.
Sure. Glucose is a single molecule, so 1 mole of glucose gives you 1 osmole of particles. But if you take 1 mole of sodium chloride, NaCl, it dissociates in a water into 2 separate ions, Na plus and Cl. So 1 mole of NaCl gives you 2 osmoles.
So the effect on fluid movement is twice as strong.
Exactly. It's all about how many particles it generates.
And tonicity takes that particle count and defines it relative to a cell, usually a red blood cell. What makes a solution isotonic?
Isotonic just means the solution matches the osmolarity inside the cell, which is typically around 280 to 300 milliosmoles per liter. This causes no net water movement, but if you give a hypertonic solution, something higher than 300, you're going to draw water out of the cells. They'll shrink.
Which is why IV fluids have to be so carefully managed.
Okay, the last homeostatic pillar here is acid-base balance.
Right. Our blood is naturally slightly alkaline. Homeostasis keeps it at a very, very narrow range, pH 7.35 to 7.45. If your blood pH drops below 7.35, that's acidosis.
And the body fights that with buffers.
It does. Chemical buffers like bicarbonate can stabilize pH swings very quickly. But for long-term, high-volume excretion of metabolic acids, you need the lungs and the kidneys. It's a perfect system integration.
So now let's make that full jump into the macroscopic physics that dictates how these systems function, starting with pressure and flow.
When we talk about pressure, we usually use millimeters of mercury, mmHg, in a clinical setting. But when you read a blood pressure of, say, 120 mmHg, you have to remember that's gauge pressure, meaning it's relative to the air pressure around us.
Exactly. It means the pressure is 120 mmHg above the surrounding atmospheric pressure, which is usually about 760 mmHg at sea level.
And gravity throws another variable into the mix with hydrostatic pressure.
A huge variable. Hydrostatic pressure is just the pressure exerted by a column of fluid due to gravity. When you stand up, the arterial blood pressure in your feet is significantly higher than in your aorta simply because of that vertical column of blood.
Which is why you measure things like cerebrospinal fluid pressure with the patient lying down.
Precisely. You have to take that hydrostatic variable out of the equation completely.
That detail is fascinating. Okay, moving to flow dynamics. Poiseuille's law comes up, and it tells us that vessel diameter is, well, it's king.
It's the single most powerful variable. Resistance to blood flow depends almost entirely on the radius of the arterioles and blood viscosity. Vein diameter is largely irrelevant to systemic resistance, but here's the key takeaway from the physics. Resistance is inversely proportional to the radius raised to the 4th power.
The 4th power. That's a huge multiplier.
It changes everything. If a major artery dilates and its diameter doubles, the blood flow rate increases by 2 to the 4th power. That's 16 times the original rate.
Wow.
Yeah. It's why tiny changes in arterial diameter have such a massive impact on blood pressure.
And the physics of breathing is just as dependent on simple mechanical laws, specifically Boyle's law.
Right. Boyle's law just states that pressure is inversely proportional to volume. When you inhale, your diaphragm contracts, your chest expands. This increases the lung volume.
Which decreases the internal pressure.
And because the pressure inside your lungs is now lower than the atmospheric pressure outside, air just rushes in passively. It's that simple.
And the actual gas exchange across the alveolar membrane is purely a physics problem of diffusion.
Entirely dictated by partial pressure gradients. Oxygen moves from the alveoli, where its partial pressure is high, into the blood, where it's low. Carbon dioxide moves the other way, from high pressure in the blood to low pressure in the alveoli. It's all passive, all down the gradient.
Okay, let's shift to energy balance. If energy intake is more than expenditure, the excess gets stored as fat. Simple enough. But basal metabolic rate, BMR, is the energy you use at absolute rest. If we link that back to what we said earlier, what's the single biggest energy consumer at rest?
The Na plus K plus AT pace pump, without a doubt. Maintaining those steep ion gradients is an incredibly expensive physical task. It consumes a massive fraction of your BMR.
And we manage excess energy by regulating heat loss. So how does the physics of evaporation make sweating so effective for cooling?
Well, cooling is all about removing energy. When you sweat, the most energetic water molecules, the ones with the highest kinetic energy, are the first to escape the liquid and evaporate.
They take the heat with them.
They literally take the heat with them, leaving the remaining water molecules on your skin with a lower average kinetic energy. And that's what we perceive as cooling.
Okay, final bit of physics. Diagnostic imaging. We hear about ionizing radiation, like X-rays, which are highly penetrating. Why is that?
Because of their short wavelengths and high energy. But let's contrast that with ultrasound. It's non-ionizing. It works on the principle of reflection, or echoes. And the key variable here is acoustic impedance.
Which is what, exactly?
Simply put, it's a measure of how much a material resists sound waves. It's density times the speed of sound. Sound reflects heavily at interfaces where there's a big difference in acoustic impedance, like going from fluid to bone. That's how we get an image.
That really helps clarify it. Okay, now that we've covered the integrated fundamentals, let's apply this knowledge in a quick, high-yield overview of some key systems.
Let's do it, starting with the cardiovascular system. Notice the structural differences in the heart. The left ventricle wall is the thickest and most powerful,
Because it's pumping to the whole body, the systemic circuit.
which offers high resistance. The right ventricle only has to pump to the low-resistance pulmonary circuit. Also, remember, hemopoiesis blood cell creation happens in the red bone marrow.
For the integumentary system, structure dictates function. The dermis is highly vascularized, but the epidermis above it is avascular. It gets its nutrients by diffusion.
Right. And for glands, sebaceous glands secrete oil into hair follicles. Sudoriferous glands secrete sweat. Simple distinction.
For the central nervous system, let's focus on the hypothalamus.
Yes. It's the body's main visceral control center. It regulates homeostasis, temperature, thirst, water balance, you name it.
And for motor function.
Voluntary movement originates in the primary motor cortex, which is in the precentral gyrus. And that nerve impulse travels down the axon using that action potential we talked about, powered by our friend, the Na plus K plus pump.
It always comes back to the pump. Finally, let's return to volume and pressure control with the renal hormonal cascade, the RAAS system. This is a brilliant integration.
It's the body's master volume management system. When blood pressure drops, the kidneys release renin, which ultimately creates angiotensin II. And angiotensin II is the critical hormone.
Because it does a few things at once.
It does three key things: systemic vasoconstriction to raise pressure instantly, it stimulates thirst, and it promotes the release of two other major hormones.
Aldosterone and ADH.
Aldosterone promotes sodium reabsorption in the kidneys, and water follows the sodium. And ADH, antidiuretic hormone, directly increases water reabsorption. It's a multi-pronged attack to restore volume and pressure.
This has been a true deep dive. But mastering the material is one thing. Performing well on a test is another. Let's look at the meta lesson here, the strategy behind multiple choice exams.
Yeah, most high-stakes exams use MCQs. And the sources here analyze a few scoring strategies. Let's look at why certain methods are chosen.
We saw that strategy 1, just +1 for a correct answer with no penalty, is flawed. It constricts the mark range because random guessing gets you a minimum score of 25%.
Correct. Then you have strategy 3. Penalize wrong answers, but leave blank answers unpenalized. This is also problematic because it rewards what's called strategic omission.
So you can inflate your score by just skipping topics you haven't mastered.
Exactly, you avoid the penalty. Which is why the most rigorous assessments often move to strategy 4, penalize wrong answers and penalize unanswered questions.
What's the thinking behind that?
It tests not just knowledge, but your confidence in risk analysis. It eliminates the reward for skipping. It compels you to answer every question and forces you to apply partial knowledge. If you can eliminate even 1 or 2 wrong answers, your odds of guessing correctly go way up, and the assessment rewards that effort.
A fascinating look at how the test itself becomes a cognitive challenge, not just a fact recall exercise.
That's the point. And the necessity of integrated learning, of knitting together all these subjects,
Okay.
is what allows you to handle those nuanced questions successfully.
Okay, let's leave you with a quick review exercise based on the fluid dynamics we discussed. A healthy adult patient is administered an IV solution with an osmolarity of 250 mosmol. Relative to the patient's blood, which is 280-300 mosmol, what term describes the solution, hypertonic, hypotonic, or isotonic, and what effect will it have on the patient's red blood cells? Think about where the water is going to move.
And as you mull that over, let's wrap up with a final provocative thought that ties everything together, right back to the Na plus K plus AT pace pump. We know it works tirelessly, consuming a huge portion of our resting energy. So if all Na plus K plus pump activity in your body suddenly ceased, which vital functions, from the action potential needed for consciousness, to maintaining fluid balance and cell shape, to even just producing body heat, which of those would fail first and why?
A perfect challenge on systems integration. Something to explore as you continue building your foundational mastery. That's all for this deep dive. We'll see you next time.